All of you, welcome to the online botany classes. Today we'll study about human genome project. As you know, the term genome means the amount of DNA present in the haploid set of chromosomes. So the entire amount of DNA present in the haploid set of chromosomes, it constitutes genome. Now each genome contains all the necessary information which are required to build and maintain an organism. So what is a genome? Genome is the amount of DNA present in haploid set of chromosome. Okay. Now let us start human genome project. Actually, the human genome project was first launched in 1990 with a goal for determining the sequence of base pairs which make up the human DNA, and also for identifying and mapping all of the genes. So it was first started in 1990. It was first launched. The program was first launched in 1990. What was the aim or what was the goal? For determining all the base pairs, sequence of base pairs found in human DNA. Okay. So that mapping of all of the genes can be done. Okay. Now let us know what are the goals of the project, goals of the human genome project. The goals of human genome project are to identify all the genes present in human DNA. Okay, most of the genes that are found in human DNA, around 3 lakhs genes are found in human DNA, and the main goal was to identify those genes. Another one was to determine the sequence of 3 billion base pairs which make up the DNA, 3 into 10 to the power 9. So first one was to identify all the genes. Second one was to determine the sequence of 3 billion base pairs that make up the DNA. Okay. And to store all this information in databases for future use. Okay. So after determining all these things, you need to store them so that they can be used in future. Okay. So these were the goals of the project. Now let us know the salient features of HGP or Human Genome Project. What are those? The Human Genome Project found out that the human genome contains 3,165 million base sequences, around 3,165 million base sequences. Okay, an average gene consists of 3,000 bases. Remember it, an average genes consists of 3,000 bases, but it varies. Okay, as it has been found that the largest known human gene is dystrophin. Okay, the largest known human gene is dystrophin or dystrophin. It has got 2.4 million bases. Got it? So total number of genes estimated in case of human being is 30,000 to 35,000. Got it? And around 99.9% of nucleotide bases are exactly same in all people. So it has found out in Human Genome Project that almost all of the nucleotide bases, almost all means 99.9% of nucleotide bases are exactly same in all people. Okay. It was also found out that less than 2% of genome codes for proteins. Okay, out of the total genome, only 2% less than 2% of genome they code for proteins. Got it? Repeated sequences that do not code for proteins are called junk DNA, which make up at least 50% of human genome. So out of the total amount of human genome, it has found out that 50% of it is made up of junk DNA, which do not code for any proteins. Got it? The repeated sequences do not have direct functions. Rather, over time, these repeats reshape the genome by rearranging it, creating entirely new genes and modifying and reshuffling existing genes. So the repeated sequences that are found in human genome actually do not have got direct functions. Rather, in due course of time or in over time, these repeats reshape the genome by rearranging themselves and creating entirely new genes and modifying and reshuffling the existing genes. Another feature that was found out through Human Genome Project is that chromosome 1 is the largest human chromosome with 249 million DNA base pairs. Okay, chromosome 1 has 249 million DNA base pairs, and it is the largest human chromosome. And Y chromosome is the smallest chromosome with least number of genes, which is around approximately 55 genes. So largest chromosome is chromosome 1 consisting of 249 million DNA base pairs, and the smallest one is Y chromosome consisting of around 55 genes. Got it? So what is the application of this Human Genome Project? First one is identification of mutation which are linked to different types of cancer. Okay, to find out the mutation that may cause to cancer. Second, design of medication and more accurate prediction of their effects. Okay, through Human Genome Project, by mapping the genes, we can design a medication and so that we can predict more accurately regarding their effects. And another one is that it will help in the forensic science and biological research. So these are the various applications of Human Genome Project. Got it? So you came to know what is genome, then why Human Genome Project was launched, and what are the various salient features of Human Genome Project and its application, in which field this particular information that we found out from this Human Genome Project can be used. Okay. So this is what your Human Genome Project. Thank you.